Kevin here with a quick announcement. If you've tried to send email to us in the last, 6 months, it turns out that the email addresses that we were using weren't actually correctly forwarding to us. So they're working now, thanks to listener Ben for pointing the error out and going to heroic efforts to figure out a contact email for us. But if you've sent feedback, we're sorry we didn't receive it. So try again now, comments@emunity.org. And the form on the website should now be functional. And of course, Facebook always works. So with that, after a little delay, here's the show.
This is Audiommunity, a podcast about our body's never-ending fight with the outside world.
Hello, this is Audiommunity. Thanks for downloading us today. I'm Kate Franz, and with me today is Kevin Bonham.
Hello, hello.
And Matt Woodruff.
Hi, everybody.
Awesome. So before we hop into the paper, what are you guys drinking today?
So last time we recorded, I was drinking vodka, if you remember. And so I'm sort of swinging the pendulum on the other direction and decided to go with tea today.
Mm-hmm. Are you sick? Is something wrong?
No. I just decided that I would eventually start coming across as an alcoholic, so it was probably best not to consume straight liquor all the time.
I see. Well, thanks for throwing the rest of us under the bus, because I'm definitely drinking a beer.
All right.
Well, I mean, I did accuse you of being a little spring break last episode, so...
Yeah, you did.
Yeah, the tea. It's the overcorrection. You're overcorrecting. Yeah. It's okay.
That's fine. It's fine. It was cold today,
Kate,
actually, in Atlanta.
You're also drinking tea.
I am. I still have my root canal, and I refuse to get drunk with a open tooth in my mouth, because that just sounds like a very bad situation. Also,
so I'm the only one drinking. Is that what you're saying?
Yeah, okay, so yes, but I will start drinking again once it's February, because I'm also doing dry January. I think I mentioned that last time.
You did,
right?
Because I read about how abstaining from alcohol for a month or so really improves your liver function. And
was that a Vox article by any chance?
So I actually learned about this because my friend had decreased liver function, and his doctor told him, don't drink for 3 months. Come back and let's see how it goes. And it worked. His liver is really good. So I was like, this is a detox, like an actual detox, a true detox. I'm just decreasing the liver toxins and helping my liver recover a little bit.
Are you worried that you're just going to replace that with Vicodin with the root canal?
I actually have not taken any pain medication.
All right.
Yeah. Well, I mean, it kind of makes sense. Root canals actually, or at least mine, was not painful, because they go in and they bleach your nerve dead. So you really can't feel anything anymore.
Oh, good.
Do they literally use bleach?
Yes.
Wow.
You can smell it. I was like,
that's gross.
Yeah, it wasn't great, but I'm no longer in pain. I can eat rice again. That was embarrassing.
Congratulations.
Soup.
That's wonderful.
Yeah.
All right, well, I guess since I'm the only one, I should actually describe what I'm drinking. So I got this, it's called a triple, but not like a Belgian triple, like triple. This is sort of a weird combination of a Belgian brew, an IPA, and a lager. So I've seen the idea of
it's like a blend. It's like a red blend
or something. I don't know. So
red blend.
So I've seen this idea of IPL before, India Pale Lager, which is sort of a weird. It's like a lager style brew, but using lots of hops, like an IPA.
Disgusting.
And then, so this is apparently a new take. I've also had Belgian style IPAs, but this is a Belgian style IPL, and they're doing a play on the whole Belgian triple thing by calling it TRIPL.
I feel like there was way too much thought that went into this.
Woo.
Maybe, but I'm gonna try it and see if it's any good. So
yeah.
Well, yeah, it's all right.
I really want to try the Boulevard chocolate raspberry.
That sounds gross.
Oh, it sounds delicious. Love chocolate. Love raspberries. Love beer. I don't know how that goes wrong.
Mixing things together that are all good is not always the best way to go. But perhaps we should move on to science. So today we're doing a paper that Kate suggested, which is weird for me because I feel like I haven't thought about innate immunity much since I defended over a year ago. And
only what your PhD is
almost
about.
2 years ago. Yeah, but I haven't thought about it in a long time.
Yeah.
So reading this was kind of like a blast from the past. But it's called Transmission of Innate Immune Signaling by Packaging of cGAMP in Viral Particles. And the first author is Matteo Gentili, and the last author is Nicolas Manel. And it looks like they're from France.
Yes,
Yeah, it's all
Nicola is
French.
from France. I met him at Toll.
Oh, yeah.
When I went to Toll meeting. Yeah, he was really, really nice and spent a bunch of time talking to me at my poster, which I will tell you, not a lot of PIs will do because they want to hang out with their cool PI friends and not hang out with lame-o grad students.
Mm-hmm.
They really don't like grad students. They think they take down the party, but whatever. But he was really lovely. And with this paper, there was another paper that reported the same observation, and that it was also published in Science. And that paper is called "Viruses Transfer the Antiviral Second Messenger cGAMP Between Cells." And the first author on that paper is Bridgman, and the last author is, well, this is going to be an interesting, just kind of first take. Ray Winkle.
Okay.
And they seem to be at University of Oxford. So both these papers came out, but we'll only talk about the one because Nicola was so nice to me at Toll. Also,
Nice. I think that's a good way to make a choice.
It's not bad.
No,
Not bad.
this one has dendritic cells, and I thought that'd be a nicer entry point for you too.
Thanks, Kate.
You're welcome.
Yeah.
Looking out.
Although I will say that my one criticism of this paper is going to be the use of the term dendritic cell. But anyway.
Yeah, that's fair. Yeah, so this paper, I guess, so what Kevin was talking about has to do with the innate immunity. And so Kevin did his PhD studying innate immunity, and my PhD revolves around proteins that are involved in innate immunity. And so we've talked about it before, but just as like a refresher. So the innate immune system, we know that we have the professional immune system as T cells and B cells. We talk about it on this podcast all the time. But there's an immune system that responds first and quick when you become infected with some sort of pathogen. And this is the innate immune system, and it's present inside all of your cells. So I like to think of the innate immune system as the innate immune system, or the immune system for your cells. So there's like the tiny one inside every single cell, and then there's a larger immune system that's systemic throughout your body.
Can I just interrupt briefly on that? So I just want to be clear. So these are not, they're often talked about, the innate and the adaptive immune system are often talked about in sort of separate terms. And it's useful to categorize that way. But I just want to make it clear that these systems aren't wholly independent. They're communicating with each other constantly.
Exactly.
And so though you might study only an innate immune response, it's worth noting that all the stuff that happens in the innate immune system is directly linked in and necessary for the adaptive immune system. And one of the main functions of the adaptive immune system is to communicate and sort of enhance or direct the innate immune system. So
do you mean to reverse that?
Do you
mean to reverse that?
No, I think it goes in both directions. Like T cell cytokines are acting on macrophages and
okay.
things like that.
Okay. Yes, definitely. So the innate immune so I study the innate immune system in the context of viruses, and I don't really care about T cells and B cells. And I think the innate immune system is interesting, independent of its role in triggering and turning on the adaptive immune system, because I'm not a real immunologist. So
true.
Yes, but if you look at the Nobel Prizes that were awarded for the discovery of the components of the innate immune system, they say the importance of these discoveries is finding the mechanism by which the adaptive immune system is activated. So it's like right in the accolades for these discoveries. They talk about the implications in the adaptive
Yeah, they
immune
just
system.
shove that knife right under the ribs of all the innate immunologists.
But
right.
I do think that it is a slightly overlooked point that the adaptive immune system is very effective at fighting off infections, or it has a very important role in blocking the establishment of infections. So if you have a very effective innate immune system, you never need your adaptive immune system.
Sure.
But
sure. And I think it's worth too, we talk again, innate and adaptive systems. And even when we're talking about innate systems, we're often talking about cellular hematopoietic immunology, right? Mm-hmm. And so I think it's also worth noting that when we talk about, because you're going to get into these innate sensing mechanisms whereby an innate immune cell can detect whether there's a virus in the area, whether there's a bacteria in the area. It's also worth mentioning that it's not, the innate response is not just restricted to the hematopoietic lineage.
Yeah, absolutely.
So we're going to probably at some point talk about how each individual cell, stromal cells, so muscle cells, blood vessel wall cells, skin cells, dermal cells, obviously not epidermal cells because they don't have a nucleus.
Yeah.
But all of these cells come equipped with their own antiviral, at least, and I assume some antibacterial mechanisms that sort of help the cellular innate immune system do their job. So, yeah, the idea of an innate system is a very broad idea, I suppose.
Especially when you think about epithelial cells and fibroblasts, which are a major cell type that are at the barriers of between, like your skin, your gut. You have these epithelial cells, you have these support cells, these fibroblasts, and these cells, because they are where you interact with the environment, is where a lot of infection starts. So when you inhale air and you inhale droplets of spit or water that have flu viruses in, the flu virus will go into epithelial cells that are like in your nasal passages. When you eat something that's a little nasty, you get infections in the epithelial layer of your gut and maybe get a little diarrhea. So these cells that are infected first also have an innate immune system and are important for curbing these infections. So you don't die from two-bucket disease, as I like to call it. Double dragoning.
Oh, God.
Yeah. So yeah.
Let me just throw one more wrinkle in here. So, what do you think about? There's one sort of layer that's even more basic, I think. So, you mentioned the idea that the innate immune system is the immune system for your cells, as opposed to the adaptive immune system, which we might think of as the systemic immune system. Mm-hmm. But there's also a sort of even more local. So, the innate immune system will, when it gets triggered, it will also signal to neighboring cells. And we're going to talk about that in this paper.
Yes.
So it will also release paracrine signals like interferons.
One of the most important
some
jobs,
cytokines.
Obviously.
Yeah.
That's how the adaptive gets activated. That's how other non-specialized cells become resistant to infections.
Right. But there's also this idea, and some people like to distinguish it from innate immune system, the idea of the intrinsic immunity or the intrinsic immune system, which is really cell specific. And so this is like things like TRIM5 alpha and things like that. How do you feel about that distinction? So these are proteins that are expressed within cells that supposedly affect infection within that cell, but they don't signal more widely. They don't have more systemic function.
I mean, so those sorts of things I call restriction factors. So restriction factors are proteins. They're actually usually interferon regulated. So they're usually turned on when interferon is signaling. But they're proteins that have evolved to block the infection of viruses. I don't know if there are any restriction factors for bacteria. I don't study bacteria, so I'm not entirely sure. But so TRIM5 alpha is a protein that prematurely disrupts the capsid organization of HIV when it enters a cell, so it can stop the infection of HIV. There's a bunch of different HIV restriction factors that have been identified. Another one that will be in this paper is SAMHD1, which lowers the available pools of nucleotides. So replication is very hard for the viruses. So you can kind of block replication. I I have a friend who for his PhD studied restriction factors, and studied these sorts of like antiviral mechanisms. And he was very adamant that it was not immunity and any form, that it's just cell biology. But I do think that it does have a place being called innate immunity, because it's something that is evolved specifically against a pathogen, evolved in such a way that the pathogen has a very difficult time evolving around it and blocks the infection. So I think when we think about the sensors of the innate immune system, like the canonical players in the innate immune system, one way that we identify them is their evolutionary role. So like the fact that they evolved to detect pathogens, and restriction factors have done the same thing. In fact, you can't be called a restriction factor until you can prove that there's been positive selection for the protein over time.
Right. I feel like any immunological mechanism is just cell biology, right? When it comes down to it. So I'm not quite sure that I buy that. And I think that we're not trying to pigeonhole it by calling it innate immunity like it's not part of any other system. I think it's really just because you talk about protein cascades too, right? So complement, which is a protein system that's set up in the blood, and it's ancient systems where basically anything that's not eukaryotic, that doesn't have complement, basically something that doesn't have the ability to cleave the complement protein off its surface gets targeted by these proteins. And, these are totally cell-free environments. So I just consider all of it part of your general innate response.
Yeah. I think I'm more interested in the idea that these are factors that have evolved to stop infections than I am that the innate immune system has to end in a transcriptional response, which some people might argue. But I personally I study non-transcriptional responses of the, of the innate immune system, so I'm very partial to saying that transcription isn't the full story. Though without transcription, you would never activate the adaptive immune system. So if you think the innate immune system is only important because it activates adaptive immunity, you probably don't give a shit about restriction factors.
Right.
Yeah, and it's also worth noting that these labels that we give to things are entirely human constructed.
Sure.
It's just fun to put things in boxes.
Right. Okay, anyway, sorry for that long divergent.
Yeah. So today, though, we will be talking about the innate immunity that ends in a transcriptional response. So signaling that ends in the activation of transcription. So, I mean, I feel like we've gone over this before, but when
it's always worth repeating.
Yeah, I mean, I almost feel like I can just go on autopilot with this. So call me out if it sounds too autopilot-y. But when a pathogen enters your cell, there are pieces of that pathogen that are molecularly distinct from your own cell. So there are patterns. We call them pathogen-associated molecular patterns. So these are components of a virus or a bacteria that are not found naturally in human cells, or mouse cells, if you're me and you care about mice immunity. But so these host cells have sensors that have evolved to detect these molecular patterns. And they've picked these patterns because the bugs cannot evolve away from them. So these are things like the genomes of viruses, the cell walls of bacteria. The viruses and bacteria are not able to mutate away and lose these components. The virus always has to have its genome. So it's not going to be able to lose its genome. So these sensors that detect these molecular patterns are called pattern recognition receptors, or PRRs. And when these PRRs are activated, they send a signal down to an adapter in the cell that then coordinates a transcriptional response. And these transcriptional responses usually, we talk about the production of interferon. And so interferon is this really great signaling molecule that can signal.
If I do say so myself.
It's very interesting. It's used therapeutically to treat quite a few diseases and infections. And so interferon is released from a cell. It can then turn around and bind the cell it came out of, or it will go and bind to other cells nearby. And when it
Call
binds,
that autocrine and paracrine signaling.
And paracrine signaling. And so when it binds the outside of a cell, this transduces another signal through the cell to activate even more transcription. And these genes that are transcribed are called interferon-stimulated genes, and they have antiviral, antibacterial properties, and they protect a cell from being infected. So once a cell that has never been infected receives the interferon, senses interferon is in the media, binds the protein, it then activates all of these, the transcription of all these genes, and the proteins are made, and these cells become pretty much impossible to infect with virus. So you can protect yourself by using interferon.
I think this is a really important point. Oh, sorry, go ahead, Matt.
So do we know how far... So we talk about sometimes the localization of immune response in vivo systems, which is something that I think people don't think about often enough. So we often talk about these antiviral states, and that's how I always thought about the interferon response, as sort of creating a localized antiviral state where the surrounding cells and tissues are kind of a little bit more resistant than they were before. Mm-hmm. Do we know, has there been a study that shows exactly how far that antiviral state extends? So do you get interferons released into the lymphatics? Do you get, I know the lymph node eventually will see signals from the ensuing innate response, but I don't know if interferon itself actually finds its way.
You can certainly detect interferon in serum. So
you think the entire organism goes into an antiviral state, or do you think that it's...
Well, I'm not sure how much interferon is getting out of the serum into other local tissue. I suspect that, I don't know if there has been the direct answer to your question, if there has been a study, I don't know what it is, but I suspect that there is a large concentration dependence to the antiviral state, to the signaling that's induced by interferon. So the local environment that is getting the most interferon production is going to get the most interferon signaling and is going to have the strongest antiviral state. But you do get systemic release of interferon, and so you can get endocrine effects of local inflammation. Mm-hmm. I don't know. Interferon itself is not a pyrogen, is it, Kate?
Not that I know of.
Okay.
I'm not entirely sure.
But anyway,
the one thing I would say, so because interferon has been used therapeutically, people have done different sort of pharmacology studies on it, and they would have looked at how deeply penetrating can interferon get in an animal, and as much as you could in a human. But I've never read those clinical trials, so I have no clue.
Therapeutic dose of interferon is probably going to be different than the actual interferon that's produced.
Yeah, I guess I'm just wondering, in a normal, surface response, how much interferon could I expect at that local place versus downstream tissue and things like that? How much of my hand is really strongly antiviral if I put it on a nail that's now wounded?
Yeah. Matt, you froze on me, so
me too.
Your response was, I'm sure, recorded, but I didn't hear what you said. But I think I have the gist.
Yeah.
I also wanted to mention, it's worth, I think, stating that this idea that interferons lead to a situation where you're very resistant to future infection. I think a question that often comes up, at least among my students, is why wouldn't you always have the antiviral signaling? If signaling of interferon induces a state where you are less susceptible to viral infection, why wouldn't you just always have it on?
Yeah, because it's not that great for your cells.
Right. So the antiviral state, it protects you from infection, and it's worth it if you have positive signal that an infection is happening. You want to turn on this antiviral state to protect the local tissue from future infection. But all of these proteins that are doing things like restricting nucleotides that Kate mentioned, or inflammation generally, can be really damaging. And this is true of most immune responses. Most immune responses cause damage, and we want to restrict them only to the times that they're necessary to actually combat infection. If you always have them on, you're going to be in trouble.
And if you do always have them on, you're going to develop autoimmunity. I almost said Audiommunity.
I do that all the time now. It's really frustrating.
There are different diseases that the characteristic is that there's a high level of interferon being produced, just completely unregulated, and the effects are pretty painful.
Sure.
It's not great...
I would point out, though, that in a lot of those studies, in a lot of those diseases, the cause effect has not really been worked out. So it could be that there was another insult independent of interferon alpha. So in mouse studies, for example, of what's the lupus model that we always used to use?
SLE?
No, I mean
that's just lupus, isn't it? No,
yeah,
is that
that's
the
just
mouse
lupus.
Model? I don't know.
I don't know. We used to use this model anyway. It doesn't really matter. You do get these high levels of interferon
you know what I was thinking?
induction.
EAE.
Yeah, EAE. It is not EAE. It's a lupus-like mouse model, but you do get a lot of type 1 interferon or type. Yeah, actually,
I was thinking. So
there's, but you can treat with anti-interferon and get relief of some symptoms, right?
Sure, but you can't. Yeah, exactly. It's a little bit unclear. It's a chicken and egg problem, but it's pretty clear that the interferon is really important in B cell responses, and B cell responses, lead to antibody production and all of those things. So it definitely feeds back on itself, but it clearly is involved.
There are some where you just have overactive transcriptional adapters. So like there's a autoimmunity where you have an active allele of sting, and so they have like all sorts of like skin rashes and kind of like gnarly pathology. And there's like the TREX1 deletion, I think, is pretty clear that that's just like an overproduction of interferon in the brain.
Mm-hmm. That sounds awful.
It's not great.
Yeah. But speaking of Sting, Kate.
Yes, Sting. Sting is my baby. Yeah, so we've talked before that there are different types of viruses. Obviously, there are many different types of viruses, but there are two broad groups of viruses, and we categorize them on the type of genome that they have. So if a virus encodes all of its proteins in a genome that is made of RNA, we call them RNA viruses. So these are flu viruses, different cold viruses, viruses that give you diarrhea or hemorrhagic fevers. A lot of those are RNA viruses. Or
dengue, right?
Dengue is an RNA virus, yeah. Zika virus, if you've been listening into the news, is an RNA virus. And then viruses that encode all of their genes in DNA are called DNA viruses. And probably the most famous DNA virus is herpes. And
with good reason.
Yes.
With good reason. Herpes is a very widespread virus.
Exactly. One of the most prevalent viruses.
You have it. Don't pretend that you don't.
Me?
You, the listening audience,
the listening
all
audience.
of you.
I've actually
Everyone.
been titered for all of so a while back when I was working with live HIV, we would get titered for HIV just to make sure we hadn't accidentally contaminated ourselves, which is lovely. Not stressful at all. And on the form, you can pick all the viruses you'd like to be titered against. So I just picked everything when I was doing these HIV titers. So I got I got my Hep B titers. That's actually to look at the efficacy of the vaccine too. I got my EBV, my CMV, my HSV1, my 2. I was like, I loved knowing all this information. And I was
But you're not going to share it with us?
I will absolutely share it with you. I was 1 and 2 negative, but positive for CMV.
Mm-hmm.
So I thought that was kind of interesting. But I mean,
I've never been titered, but I get cold sores all the fucking time, and it's really irritating. So
yeah, I've never had a cold sore.
I hate you.
Yeah, I'm sorry. I mean, I'm a woman, so I have other things that bother me from time to time. So let's just call it even.
Okay, fair enough.
Yeah. Okay, sorry. I was taking a sip. I was taking a sip of my tea. So RNA and DNA viruses. So there are different pattern recognition receptors to recognize RNA viruses and a different pattern recognition receptor to recognize DNA viruses. So the DNA recognizing pathway is the most elegant, beautiful pathway, cGAS and STING. And so cGAS is a sensor that directly binds to double-stranded DNA And when it does so, it synthesizes a small molecule that is a cyclic dinucleotide. Okay, so this is the sickest thing that happened during my PhD time, was the discovery of cGAS and the cyclic dinucleotide cGAMP.
Yeah, this is only like a couple years old, right?
Yeah, this
I
came
feel
out-
like there's been an explosion of these papers.
Christmas of 2012. It was
Kate has the date in her diary.
I do. It came out like December 21st, 2012, because it was like all the ones and twos. So cGAMP is a cyclic dinucleotide, and it is the first cyclic dinucleotide identified in metazoan species. Usually, cyclic dinucleotides, we see them signaling in bacteria. And so it's really important for different bacterial processes. But this was the first one that was identified in metazoa, which I thought was really cool.
So we should say what this is. So people have probably heard of like ATP as the energy source for cells.
Yes.
But that A in ATP is the same A that is used in like DNA and RNA.
Yes.
So it's a nucleotide, and the triphosphate form, that's the TP in ATP, is just the energy bound form. And you can also make diphosphate and monophosphate versions of these nucleotides. And there's also, DNA code is A, T, G, and C. There's also CTP and TTP and GTP, and all of these. GTP is used in a lot of other signaling processes. The other triphosphated nucleotides are mostly used to construct more DNA. But a cyclic dinucleotide is essentially where you take two of these nucleotides and you hook them together covalently so they form a circle. They form a closed loop.
Yeah.
And maybe we should probably put a picture of this in the show notes or something.
Yes.
But there are other cyclic nucleotides that people may have heard of, like cAMP is a second messenger that's taught in a lot of biochemistry courses. So that's cyclic adenosine monophosphate. But that's a single nucleotide that's covalently bound in a loop. This is so cGAMP is an AMP, so an adenosine monophosphate, and an AMP, so an adenosine. Wait,
GMP.
did I do that right? GMP. Thank you.
We should definitely have a diagram of these.
Yeah, and so it's two nucleotides that are stuck in a loop. So,
and they're kind of hooked onto each other. It was a big controversy how these things were hooked onto each other when cGAMP first was discovered. Everyone thought that it was a 3', 3'. So 3' just means the third carbon in the sugar. The third carbon in the sugar was linked to the phosphate group through a phosphate linkage to the second carbon of the second sugar, or the third carbon of the third carbon of the second molecule, but
of the fourth,
just like anyway. We thought that they looked more like bacterial dinucleotides, but now when we look at the linkages, we see that it's actually slightly different, and it actually leads to different signaling. If you see the different linkages, they signal slightly differently. Anyway,
so can you make a cGAMP that is linked in the more bacterial way, for lack of a better phrase?
Yeah,
and it has different effects.
Well, if you have the bacterial type cGAMP, it will signal in mouse cells. So the receptor, I should say, the receptor of cGAMP is the protein STING, which is one of my favorite proteins. And
do you know what STING stands for?
STING stands for Stimulator of Interferon Genes, which is a cute little acronym.
Yeah, but that's kind of a crappy acronym because it's always done in all caps, and I feel like it should be, if that's the case, I didn't know that. But if that's the case, I feel like it should be capital S, lowercase t.
Let's not pretend that these acronyms are well thought out or implemented.
That, if we do it, it's going to, every time I write a paper, it's going to look like I'm actually writing in my live journal.
Right. Okay, fair enough.
RIP live journal.
I feel like the acronym world has to undergo what the cluster of differentiation molecules did however long ago, where we just need to get rid of a lot of these things and start numbering them.
You will
No.
take my
No,
proteins
Kevin-
and replace them with
Kevin
numbers
disagrees.
over my cold dead body. Like, if that ever happens, I am going to defect into flies full time, because flies have the coolest gene names, and they will never go to stupid numbers because those people are nuts.
They are
And
nuts. The
probably
CD
nuts.
the CD,
Don't
the
take
cluster
that
of
as a bad
differentiation
thing, Lori,
thing
in our
is
lab who works on flies.
yeah.
the cluster of differentiation thing is a pain in the ass, and I hate it.
Yeah, but you have numbers that actually correlate to proteins in people's minds, right? The problem with these acronyms is we've gotten so far that the acronyms now start referring to previously held acronyms. And so you can get into the acronym and come up with another acronym. And also,
I'm well
not
aware
have
of this.
to- it's ridiculous.
I did this as an exercise one time, like on my blog back in 2010, and I took a 3-letter acronym that expanded out into like 15 words.
Yeah, that's absurd.
I mean, it works.
Okay. Yep.
Okay.
I'm sorry. When you have a Nature paper that has both the word ninja and the word topless in it, and you can publish that in the highest impact journal in the world, I think that's better than CD whatever the hell.
I mean, it is clever. I'll give you that. It's clever. But it's only sort of clever at this point. Also, I'd like to try to reinstate a moratorium on all talks with colons in them. I was recently given another pamphlet. I don't want anyone out there listening to put together talks that have colons in them.
I think that's really
Are you
rude to
talking about the end?
gastroenterology.
Yeah.
Yeah. No, actually,
There will be no more talks on my floor.
it is. It is actually a mouse intestinal talk.
So it's got both kinds of colons in them.
Yes. Yes. Correct. Sorry.
I think so. Normally when I edit this podcast, I have my audio editing software delete long silences. I feel like in this episode, I'm going to need to leave in these pregnant pauses because I think
there
it's
have been a
important
couple of
for
them.
As
be-
we think
be
about
more
what
selective.
we've done.
Yeah, exactly.
Okay. Back on topic. So, DNA.
And I'm the only one that's drinking in this podcast.
Viral DNA enters the cell. cGAS binds the DNA. I am oversimplifying this a bit. There are regulators and other things, but we don't care about them right now. cGAS binds viral DNA, makes the small molecule cGAMP, and cGAMP travels to STING. STING is located in the ER of a cell. Travels to STING and binds STING, and STING is activated. It recruits transcription factors, and interferon is turned on. Okay. Super cool. So HIV, when it is infecting cells, it's kind of an interesting virus in that its genome, when it enters the cell, is RNA, but it's quickly reverse transcribed into DNA. So we kind of think of HIV, at least I do, from an innate perspective as a DNA virus. So when HIV makes its DNA and then the DNA is then inserted into the host genome, that DNA can be either that DNA that's being inserted or abortive transcripts of this DNA activate cGAS. And that's probably one of the more important things to know about this paper before going into this paper. Is that
when you say abortive transcripts, do you mean abortive reverse transcripts?
yes.
Oh.
So
it never even occurred to me that was a thing, although of course it makes sense that 100%. It is, but
100%. Yeah. So when the reverse transcription is occurring, you can have an abortive event happen where you just make some of the DNA and not all of it, or like halfway through you run out of nucleotides, or there's like an error catastrophe and everything just goes to pot. Like there are a lot of events.
Goes to pot. I really like that phrase.
Yeah. I thought I would try to lower the S-bombs.
There's already been multiple curse words.
Yeah,
I marked them in my audio, so I know to remove them.
So I thought I'd block the shh that's going to occur. Can I ask a question that I know that I should know the answer to this, but I don't. Okay.
So I'm going to ask it. So HIV has the RNA, single-stranded RNA genome that's reverse transcribed into a single strand of DNA.
No. So wrong on both counts, just from the get. So it's actually a dimer of RNA genomes. So it's not double-stranded RNA. It is single-strand RNA, but it's a dimer of the genome. So they're actually bound together. There's base pairing at certain parts throughout the genome.
Okay, but they're not complementary strands. They're both the same strand.
They are not complementary strand. They are the same strand. Yeah.
Okay, so just for simplicity's sake, let's say we have 2 copies of one strand.
Yeah.
Of the genome, and the first thing that happens is it gets reverse transcribed into a single strand of DNA.
Yeah.
So how is the second strand? How is the complementary DNA strand produced?
So what happens is that you make the first strand of DNA, and then you flip around to the second part, to the RNA DNA hybrid, and you start chewing away the RNA to use that DNA strand as a template for the second strand of DNA.
And the reverse transcriptase is doing this as well?
Yes.
Okay.
Yeah, so the reverse transcriptase has RNase activity, so it can digest RNA, and DNA polymerase activity, so it can actually make strands of DNA.
I just taught an extension school at the virology course, so I should really know this, but the good news is that Jamie gave the lecture
for
the HIV. So she took care of that, and I was not paying attention apparently.
So I really oversimplified it.
Sounds vaguely familiar.
Yeah,
but
I did oversimplify it. There are a bunch of different there's primers involved. Obviously, if you have learned about transcription or the DNA synthesis, there is a little bit more, and it's kind of confusing. It's a 9-step process, but
super cool molecular biology, but we're going to skip it
essentially,
for this podcast.
You use the template RNA for DNA strand, and then it gets digested, and you use that DNA strand that's left over as a template for the second strand.
Cool.
Yeah. And you can, so because there's so many steps, you can lose the transcripts at the actual synthesis can stop for a number of different reasons, just in the regulation of it. Yeah, but take-home message is that the HIV DNA activates cGAS, and that has been in a couple of different papers now. So, now that we've explained the background for an hour, we'll get to the paper, which is well worth it. I think that this paper was one of my favorite papers of 2015, in that the idea behind it was really cute. It was quirky. You wouldn't exactly have expected it. So
remember from a couple episodes ago when Kate says cute, that's not that's cute, sarcastic cute. She actually means it.
It's cute. It's quirky. It's an interesting observation. I wouldn't say. So that's the thing I go for when I like something, but it's not the most elegant thing that's ever been laid out. It's more of the observation, and then when it's something's improved really interestingly, then it's more elegant. Mm-hmm. So that's where these are where my compliments break down.
Fair enough. The thing that I thought was really interesting about this paper is it seemed like the way that they wrote it, which is the way that a lot of science happens, but people rarely indicate this. It sounded like they were trying to look at one thing,
yes,
and actually figured out something else.
Exactly.
And they wrote it to demonstrate that, which I think is cool, because I think that people there's a lot of serendipity in science where we think we're looking at one thing and we find something totally different and then run with it.
Yeah, exactly.
But I feel like when we write up the manuscripts, we typically pretend that we had the idea all along.
Yeah.
But so it's really nice. It was really refreshing to read the thing where they're like, yeah, so we wanted to do this thing, and then we saw this other thing, and turned out that was really cool, and we got a science paper.
Yeah. Yeah, definitely. That's the other reason why I wanted to focus on this paper. Is it almost reads like someone's lab journal.
Right.
I did
it really
this,
does.
I had no clue what the hell was going on, so I did this other experiment. I think I'm probably wrong. I probably have messed all this up, but I did the second experiment, and everything is still corroborating this weird idea. And here it is.
Yeah.
So I have to say, I sort of agree with you guys in that I think that it is interesting because it does sort of display the way that science is often discovered. But I think part of the purpose of putting together a final manuscript is to make sure that the reader is walked logically through the proof of what the thesis is going to eventually be. And I did eventually think that this paper got there. I totally bought everything that they said at the end, but when I was going through figure 1, I did not know where the paper was going to go, right? Because if figure 1 is, hey, we did this experiment and look at this weird thing that happened, right? I'm no closer to understanding what's going on.
I agree.
So I understand what you're saying. I do like being able to see what the thought process was, but I thought that reconstructing this could have been more useful as far as guiding the reader through the paper.
Yeah.
Yeah, maybe. Although it kind of reads, I mean, this is a gross oversimplification, but it kind of reads like a mystery novel. Like you get presented like in the first figure with like what the hell is going on here? Which I think, I mean,
yeah, no, I totally agree with you. It definitely does read like
watching shows like Sherlock,
Bones,
and no, not so much, but okay, but like shows like that where you're presented with a mystery and then it sort of goes through and then you figure out the mystery at the end.
Law and Order SVU.
But I never find that with science, and I think it's fun to have it sometimes.
So it's a cross-genre piece of work, is what you're saying.
Yeah.
Okay. Yeah.
So the initial observation that upset Matt, I actually
it did not upset me.
I'm joking.
Yeah. Well, maybe I should just, before we even go into the data, I should just say that the immune system. The adaptive immune system is great in mice because you can pull out B cells, you can pull out T cells, you get tons of them out of any tissue that you want, really. You can find a way to get enough of them where you can do an immense number of studies. It gets a little harder when you're talking about innate cells like dendritic cells and macrophages, especially if those dendritic cells or macrophages have to be tissue specific. The problem with these cells is that they're incredibly diverse. So we talk about dendritic cells a lot in this show on Audiommunity. But realistically, from each individual tissue that you want to pull from, if you're looking at the skin, for example, you can probably pull 5 different subsets of dendritic cells out of the skin. And all of them look pretty different from each other. They certainly have their commonalities, but they perform different functions, they respond to different things, right? And your skin-resident dendritic cells are not going to look the same as your lymph node-resident dendritic cells, which certainly don't look like your peritoneal dendritic cells. And so all of these things have their own, specializations within them. The problem is that it's really hard to get any number of these cells out. So we can do flow cytometry, we've talked before, and see that these cells exist, for example, in the skin, but it's really hard to get a good population of dendritic cells out of the skin in enough numbers where you can then do functional experiments on them. So one of the tricks that immunologists have used in order to sort of try to get around this is it turns out that you have large monocyte populations that are floating around in your blood at steady state at any given moment. And it's obviously very easy to get the blood. So if you bleed an animal and you have these monocyte precursors, and some monocytes have the capability of differentiating into DCs, what you can do is you can put all of this blood tissue into a plate with the correct factors. So these are dendritic cell propagating and differentiation factors, right? And so the idea is that you're starting from a precursor and you're differentiating these cells in vitro, and what you get on the other end is a cell that looks like a dendritic cell. And so in mice, and I'm a little hesitant to talk about humans because I don't know the literature well enough, but I believe that there was a paper that just came out expressing some of the same issues. In mice, we have overwhelmingly in immunology used these monocyte-derived dendritic cells in vitro to test really complex ideas. And it turns out that the monocyte-derived dendritic cells that we get, first of all, most labs, when they're doing these experiments, aren't actually using dendritic cells.
Yeah.
So this paper basically suggests, first of all, that the majority of labs that are using these monocyte-derived dendritic cells are probably not using dendritic cells at all. So if you do transcriptional analysis on the cells that they're using for these experiments, what you find is these presumably pure dendritic cell populations are actually heavily contaminated with inflammatory macrophages and monocytes, which basically, I mean, it's going to skew your data heavily. We talk about these dendritic cells.
That's totally true. Sorry to interrupt, but I totally understand what you're saying, but for this paper in particular,
yeah.
the etiology of these cells doesn't actually
matter. So the only thing that I would say here on the front end of this paper is that I totally buy the vast majority of what's being shown as far as, transfer of these small molecules in a viral vector. But we'll get there. Spoiler alert. But just be careful in how you interpret the cellular response to these things, because the models that they're using don't necessarily indicate a dendritic cell.
Right, and they move away from dendritic cells also fairly quickly, but so that warning is definitely worth heeding. But in this particular case, the nice thing about studying the innate immune system, I think Matt is showing his bias as a person who cares more about adaptive immunity, but the nice thing about the innate immune system is it turns out that a lot of these systems work fundamentally the same way in a lot of different cell types. And so we can study different cell types and have different output, like different readings for activation of pathways. But when you care about the activation of the pathway, it sort of doesn't matter if you have a pure population of well-differentiated DCs. All you care about is saying, like, hey, we can measure co-stimulation, which is a measure of activation.
So let me throw this.
We're in good shape.
So here's my actual concern with the interpretation that these are actually DCs and they would act like DCs in vivo. I'm not convinced that a DC in vivo would be able to pull in these particles in the same way that a highly phagocytic inflammatory macrophage is, which is, I think, what they're probably dealing with. And so I would
that doesn't actually matter in this case.
Well, it does if you're talking about.
So it actually does matter for HIV.
Yeah, exactly. Yeah. For the problem of HIV, it does matter.
Yeah. I mean, the idea that HIV directly infects the dendritic cell, and that is how that's how like the activation occurs and how the immune response starts, I would say is a little controversial.
Okay. That's fair. I guess that wasn't the exciting part of this paper.
It's definitely not the exciting part of the paper.
No,
it's not.
It's not
necessary
and it doesn't
to
invalidate any of the main conclusions. It's just an important point in the interpretation of what it means to HIV and to similar diseases.
Yeah.
Fair enough. I need to run. So I'm going to sign off here. I trust you two to go through the content of the paper.
All right.
Yes.
So I will see you guys later.
All right. We'll see you later.
Okay. Bye.
Okay. So initial observation is that so they made two different types of HIV virus. So these aren't encoding HIV proteins. They actually encode just GFP or just cGAS. So it's a technique to be able to express transgenes. Is you've got the inside of the HIV genome, you express all of the components of the virus in a cell with the genome that has your gene of interest, and it will package it to look like HIV, but you will not have any HIV parts inside of that virus. And so they're called lentiviral vectors, and it's a really good tool that pretty much everyone uses to get expression of your genes. So when they were making these viruses and then infecting their maybe DCs with them, they saw that the viruses that encoded cGAS activated DCs, whereas a virus encoding just a fluorescent protein as a control did not. So they probably were just trying to make DCs that express cGAS to study something about cGAS. But they got this interesting phenotype between the two different viruses.
So can I ask you a quick question? Have you worked with lentivirus before?
Oh, yes.
Do you know what the capacity is? Like, how many genes can you insert in these things and still get good expression?
So your expression goes, so your virus titer, so essentially the length of the genome will determine how much virus you get out. So the longer the genome is, the less virus you get out and the less infectious it will be.
Okay. So it's just a trade-off.
Yeah. So you can get great expression off of like 2 kb of insert. And then every kb you go past that, you probably lose like half a log of infectivity.
Okay. So there is a limited capacity.
There is a limited capacity, but these are well within.
Sure. Yeah, that was just a totally technical question. I just have never worked with the vectors.
Yeah. They first thought, so the first like reasonable hypothesis that you could make is that cGAS overexpressed will just generate a lot of the second messenger cGAMP. So they thought maybe cGAMP or cGAS is inserting into the genome, expressing, and then that is how the activation is occurring. So they use this trick where if you use these viruses in the presence of this viral protein VPX, which is from SIV, it will counteract one of the restriction factors, and it will make a more efficient transduction. So you'll actually have a productive infection. So they remove VPX from the system, which will basically lead to the very inefficient integration of the DNA. So when you remove VPX, you are eliminating basically the cGAS gene inserting and being expressed. And they thought if it's just from cGAS, VPX will eliminate all the taking out VPX will eliminate the activation.
Okay, yeah. So this lentiviral construct basically at steady state, before you start modifying it, contains this VPX gene. Is that reasonable? That allows the insertion of whatever gene you put in there into the transfected cell?
Yeah, essentially you can make the virus in the presence, so it will carry the VPX protein with it.
Got it. And so by eliminating VPX from some of these viral particles, you're introducing the gene, but you're not allowing that gene to integrate, and so it can't be transcribed, and so you're not actually getting cGAS production in these cells.
Exactly. So when they infect with these viruses without VPX, the dendritic cell is still activated. So they had the idea that perhaps, in the production of these viruses, when you transfect in, when you have in the viral genome, you have all the virus pieces in that producer cell, the cell that makes the virus. They thought perhaps it's grabbing something from the producer cell and incorporating it into the virus. And that is what is activating the dendritic cell. So to test that directly, they made their virus again, but they made virus that didn't encode cGAS, but cGAS was overexpressed in the cell, or they made a virus that did not have any cGAS overexpressed in the cell. So they thought maybe cGAS or something that is activated by cGAS or something that cGAS makes is incorporated into these viruses, and that is what activates their dendritic cells. And so when you make virus in the presence of cGAS, those will activate the dendritic cells. So that told them that there was an innate signal present inside of those viruses.
Yeah, so this is cool. And this, again, it gets back, it's sort of a technical component of how these viruses are made, right? There's not an initial progenitor virus that you then propagate. What you do is you take cells and you transfect them with the material that encodes for the entire lentivirus. Is that correct?
Yeah.
So basically you get the creation of this virus from a cell line from nothing, just by inserting the genetic material. And so what you're saying is, even if that virus does not have cGAS, but you previously turned cGAS on in the cell that produced the virus, then that virus will become stimulatory to the cells that are receiving it. And that's really cool. Like, imagine how frustrating that would have been to try to figure out as a grad student. Like, you're working with these viruses and you're knocking out cGAS and you're like doing all of these things, and every time you put your new virus onto cells, it still activates.
Yeah.
I can't imagine how many times that experiment was done.
Yeah, so they did a couple more experiments to show that it's the actual virus that has the signal. So they do differential centrifugation of the supernatant. So they can isolate things that are in the supernatant of the producer cells of different sizes. And they see that the activating signal always stays with the fraction that has the virus present. And then they play a couple more games with the virus, and they show that if you don't have the protein that is necessary for the virus to enter a cell, you no longer get the signal. And if you overexpress cGAS that isn't active, you again lose a signal. So they show that you need to have a virus. It needs to be able to fuse so the virus contents can get inside of the cell. And you need active cGAS for the signal to propagate, which all kind of points to this idea that there's something inside of the virus that is activating the dendritic cell. And they actually do this other experiment that I liked. It's a more HIV-centric experiment. using a different restriction factor. So the expression of this protein tetherin inhibits budding of HIV. So if you Google this, tetherin and HIV, you can look at EM pictures and you can see this protein is physically holding the virus particle in, like, from leaving the cell. So like the whole particle is almost budded, but there's this protein just anchoring it to the cell. And so if you make these viruses in the presence of tetherin, so now like the virus cannot leave the cell, you also lose a signal, further pointing to the virus holding something that's activating inside.
Is that the same protein that's responsible for those big, long T cell tethers? Where you get...
I do not know.
So there's this interesting, I think Thorsten Mempel published a paper a while back, basically showing that if you've got T cells in a lymph node that are infected with HIV, they form these huge filaments. I mean, sometimes I think a couple hundred microns, you know, where a T cell is probably 10 microns in size, but then they get these huge long tethers to each other over huge amounts of space. And I wonder if it's the same protein.
I would guess it's either the same protein or some sort of virus synapse.
Yeah.
Something where like the virus proteins are fusing the cells together. But yeah, so they still couldn't determine whether or not it's the actual presence of active cGAS or cGAMP. And so
this is whether it's the enzyme that creates the mediator or the mediator itself.
Yes,
Right.
exactly.
Okay.
So I mean, the easiest way to get around this is just to look for the presence of cGAMP in these particles.
Yep.
So basically, it's a really simple experiment. They just isolated their viruses, they lysed them open, and there's a really cool thing about cGAMP is that it's a small molecule. So it's heat resistant, it's resistant to DNases, RNases, proteinases. So you can do all these treatments to an extract and essentially inactivate all of your different PAMPs, all of your different signaling molecules. You boil them, you spin them down, and you'll be left with this extract of just dinucleotides. So they tested to see if the extract was still immunostimulatory, meaning that cGAMP is present there after they do all of these, just throw this extract through the ringer. I do this all the time. And then they sent that extract out for basically identification through mass spectrometry, and they see the presence of the cGAMP when they do mass spec on it.
Yeah.
Do we want to explain mass spec?
No, but I think
Okay.
I think it's a good public service announcement for people in biology. I think biochemistry and chemistry in general scares people away in biology because they remember organic and things, and they get afraid of things like mass spec and NMR. And I would just encourage people, this mass spec experiment is cut and dry. It's just done, right? And you could, from a biological perspective, have answered this in a number of different ways, and none of them would have been as clean as just running it through a mass spec. So if you don't know how these things work and you're a biologist... It is a really difficult
mass spec experiment.
It is a hard
one.
But you should be able to know what mass spec roughly helps you answer, NMR and things like this, and not be afraid to employ them, because oftentimes it is the simplest and most straightforward way of doing it.
Yeah, it's a yes or no.
Right, exactly. Is it there? Yes, it is. Okay, moving on. Yep.
So essentially what they show is that when a virus, what the hypothesis is that when the virus infects a cell, and it activates the immune system, not only do you secrete interferon from that cell to warn the other cells, the viruses that leave have the potential to package cGAMP into the virus, and then when they infect a new cell, they now have this little alarm bell with them. So it's essentially like putting a cowbell on a cow. The cell, before anything happens, the cGAMP activates the sting, and you can get this antiviral response and hopefully beat the virus before it replicates.
I like that in that metaphor, the cow is infectious. like someone's really afraid of cows, and so you put a bell on the cow, and then that person is warned that the cow is there.
Exactly.
Yep, I really like that metaphor a lot. So go ahead.
I was going to say, there are a couple, and so then they also looked at two other DNA viruses, mouse CMV and MVA, which is related to vaccinia. And they also found cGAMP present in these cells, or in these viruses, when you produce them in the presence of cGAS. So both of these viruses have DNA genomes. When they replicate, they theoretically will activate cGAS. And then cGAMP is made and can be packaged into these viruses.
Yeah. So I was reading this. First of all of that's really cool. I like this idea that the cell can just sort of produce mediators, right? Pro-inflammatory mediators. And because the cytoplasm is getting packaged basically into the virus, like all of those danger mediators are also going to get packaged into the virus, which I guess theoretically makes sense, but I would never have come up with, the idea that that's actually happening. But it does make me wonder, because cGAMP is not the only small molecule mediator, right? So there are pro-apoptotics.
Oh, okay. Yeah.
Right? So if a cell is undergoing apoptosis, you get cytochrome C release from the mitochondria, and now that's in the cytoplasm. Presumably, cytochrome C could get packaged into a virus as well and then activate innate proteasome.
Cytochrome C is significantly larger than cGAMP.
Is it? Is it that much larger? Oh. Mm-hmm. Oh, yeah. I'm totally screwing up my.
I mean, cGAMP is very tiny. It's the size of 2 nucleotides.
No, I assumed that cytochrome C was also very small, and I must be. Yeah, it is
small. It is small it is small, but it's not quite that small.
No, it's not.
It's a pretty complicated molecule. Yeah. I think you can actually run it off on a protein gel.
Yeah. Yep. So
I think it's like
cytochrome C
10 kD maybe.
Yeah, is a bad example. Actually, that is a protein. For some reason, I thought cytochrome C was a much smaller.
Well, it has like that tetra. What is that? What is that word? Sorry, like heme-like. I couldn't get the actual chemical word to pop in my. I can spell it actually, but I couldn't say it. But it has like that heme-like molecule in it, which still is pretty. It's like probably 5 times the size of cGAMP.
So let's drop that one. But how about things like lipid mediators, pro-inflammatory lipid mediators might be looked at. Uric acid is very small, and we know that inflammasomal activation can come. Exactly. So it does make me wonder how many groups now that this paper have come out are looking for other pro-inflammatory mediators that are being packaged inside of viruses. Yeah,
maybe small little something to activate the inflammasome. That'd be great.
Right, exactly. So maybe we can get a siren on the cow in addition to a bell.
It's like basically just loading a shotgun and taping it to the back of the cow.
Wait, now the cow is more dangerous.
No. It's okay, we just got dark, but it would theoretically kill the cow.
Oh, yeah.
Quickly kill the cow.
You went in a different direction there than I did.
What was that movie with the Javier Bardem? I was gonna say There Will Be Blood, but that's not it. No Country for Old Men. Okay. It's like the cow, the air gun. Yeah. That's for killing cows. Yeah. For those of you who didn't grow up on a farm.
This metaphor fell apart. Although I did like it right up until the shotgun, I think. So that will be my cartoon for the week.
Yeah. I will say that the one caveat with this paper is that so they make their viruses in the presence of cGAS that's being overexpressed. So cGAS doesn't have to be activated. Viruses encode lots of different proteins to try to turn off the innate immune system. So proteins that would cleave cGAS or block it from activating. So in a natural infection with a fully functional virus, these viruses could potentially turn off cGAS, stop cGAMP from being made, and then cGAMP can no longer be loaded into a particle.
That would be an awful experiment. I'll bet, because it's a science paper, right? I'll bet that they were asked for the production of an experiment that would show you that in vivo it is packaged, but that would be awful.
I assume that's why they use MVA. It doesn't really replicate. It doesn't replicate in like human or mouse cells. You have to propagate it, I think, in chicken cells, just because it's such a puny, weak virus. Please don't strike me down, Vaccinia. I'm talking bad about it.
Yeah.
I work with viruses all the time, so I kind of feel like they have personalities. So I don't want to anger the whims of them.
Don't want to anger Vaccinia.
But it is like kind of a weak ass virus.
Okay. Okay, so this may not be something that the immune system or the virus, has not thought about and countered, is what you're saying.
Yeah. So if you want to stop the intracellular signaling from occurring, you could target STING, so there is no interferon made, but it's very easy also theoretically it could be easy to target cGAS as well and stop all the cGAMP production.
I also wonder, could you have a sequester in a virus? Is there a way that you could sequester those small molecules in a virus that was a major problem? Now, I'm trying to think of all the ways I could think of for a virus to prevent the dissemination of that mediator from one cell to another.
So, yes, you could sequester. Many viruses replicate in replication factories, and they can be membrane bound. And the idea behind that, it's a little theological, but the idea is that these complexes are membrane bound, not only to increase the enzyme kinetics of the actual virus replication, but also to evade host sensing. So if you never showed your DNA to the cell, so cGAS could never touch it, you could sequester yourself off in the cell and prevent any sort of cGAMP from getting in or cGAMP from ever being produced.
Yeah. Well, anyway, I'm sure that somebody will come through and look at all the ways that, this process is disturbed in real viruses. Is that what you said before? I think I heard you say real viruses.
So a lot of viruses we use in lab are attenuated for safety and, so we don't start pandemics like Vincent Racaniello said we might have done with flu once upon a time.
Yeah, oops.
Oops.
Sorry, everyone.
Yeah. So it's nice, and it's nice to not have to get titered for your viruses every 3 months to make sure you can accidentally infect yourself.
Right, but it seems like it's fun. You said it was fun, so maybe more active viruses in labs. Yeah. All right. Well, why don't we finish it up there then?
Yeah, sounds good.
Great. So this has been Audiommunity. Kevin has left us, sadly, for this last portion, but I'm Matt, and with me again is Kate.
See you guys.
And we are around on Facebook, I believe, and we have an RSS feed open. I don't know what I'm talking about. I'm the technologically deficient one in the group, so I'm just going to refer you to other episodes that I'm sure you've heard at this point. If not, go listen to some other episodes. And until next time, watch out for the cows with bells and sirens. See you later, guys.